Hello everyone. This is going to be the start of chapter 12 in which we're going to be exploring the nervous system. So the nervous system, I think when many people hear that term, they think of the brain, they think of neurons, they think of the spinal cord, and all that is fantastic. So what we'll do in chapter 12 is we'll explore those concepts, and along the way we're also going to take a look at exactly what goes into the communication pathway, because the nervous system is an excellent way that our body is able to communicate with both external and internal stimuli, and then of course it's able to communicate with the sensory system and come up with any type of effect or outcome that needs to be done. So things like your reflexes, you put your hand on something hot, you withdraw it. This is all of course meant to keep the body nice and safe. It's part of our homeostatic settings. Now, within the nervous system itself, it turns out that we can have two major subdivisions, and these are going to be important to understand as we proceed through chapter 12. They are going to be the central nervous system and the peripheral nervous system. And go ahead and take a look at the illustration that's on the right-hand side. And what you're going to go ahead and see is that in the pinkish color, they've highlighted the fact that the central nervous system, the CNS, is going to be the brain as well as the spinal cord. So anything that's happening within the brain and the spinal cord is going to fall under the central nervous system. In fact, this is going to be where your processing centers are going to be, because the brain is ultimately going to be the one that kind of receives the stimuli and figures out what needs to get done, and then goes ahead and directs the message out into the body. The peripheral nervous system, the PNS, that is going to involve anything that is outside of the brain and the spinal cord. And in fact, on our drawing, it's illustrated as the yellow lines. And you're going to see that they've highlighted for us the nerves and the ganglia. So what exactly is a nerve? Well, it is going to be an axon that basically reaches outside of the spinal cord. So remember when you look at a neuron, because we've slightly mentioned them in our chapter 5, as well as some other chapters, when we talk about like the neuromuscular junction, we talked about the fact that a neuron cell is the main cell of the nervous system, and it has all these dendrites that are able to receive information, but then they only have one axon that sends off a signal and goes over to the effector organ, right? Okay, so what we see happening here is that these axons, when they come out of the spinal cord, they are going to be called nerves. So a nerve is nothing but just an extended axon that has left the central nervous system, meaning it has left the casing of the brain and the spinal cord. So now it falls under the peripheral nervous system. You will also have ganglia. Ganglia are going to be these swellings. You can see them on the illustration. They're like these little yellow knobs. Those are going to be the cell bodies of the neurons that are in a nerve itself. So once again, here we have neurons that are outside of the brain, outside of the spinal cord, so they are going to be part of the peripheral nervous system. Another way that we're going to be able to discuss the nervous system is by taking a look at its division between what we like to call its sensory and its motor division. Now, the sensory and the motor divisions are only coming into play when we take a look at our peripheral nervous system. So do me a favor, take a look at the flow chart that's on the right-hand side. And in blue, we can see we have our central nervous system, and we have our brain, we have our spinal cord. All right, so notice how there's no further subdivision. It's just those two particular organs. And I want you to think of the central nervous system as my control center, okay? That is where all the cells are that are going to make all the decisions. Now, how do they know what needs to be done? Well, they're going to rely on the peripheral nervous system. Remember, the peripheral nervous system is what's radiating out. Those are your nerves, those are your ganglia. So they are monitoring your entire body. And what we see happening is that your peripheral nervous system will have a sensory division. So it will be able to sense any type of stimulus, like what's happening on the outside and the inside of the body. And it will have a motor division. The motor division is the division that will actually do the work. So, hey, does your hand need to be pulled away from the hot source? Do we need to start running? Do we need to produce an enzyme? Anything that will entail the work that needs to be done will fall under the motor division. So here's how it's going to work. Your sensory division, so the one that's receiving stimuli, is going to be divided into a somatic and a visceral sensory division. You can see it right here. Visceral sensory division and somatic sensory division. Now, somatic basically is going to entail that we have receptors that are monitoring the skin, muscle, bone, and joints. So if there's anything wrong with the skin, maybe you cut yourself, if a muscle has been torn, if a bone has been fractured, if joints are being popped out of place, these are going to be registered and are going to be detected by the somatic sensory division. The visceral sensory division is going to be all about your internal organs, because viscera just means internal, right? So this is going to take a look at your different cavities. Is something wrong with your lungs in your thoracic cavity? Is something wrong with your stomach, your small intestines, your large intestines, your pancreas, your liver? That's all the viscera. And because it's monitoring the stimulus, it's going to be the visceral sensory division. So are your lungs congested? Is your stomach empty? Does the liver need to produce extra bile? These are all things that are being detected by the visceral sensory division. The somatic sensory division, once again, is more external, taking a look at the skin, the muscle, the bone, and the joints. So they are the ones that work together with our sensory system, like our eyes, our ears, our nose, our sensation of touch. They work together, and once they receive a stimuli, the sensory division will send that signal over to the central nervous system, where the brain and the spinal cord, mostly the brain, will receive the sensory information, and it will figure out what needs to get done. Hey, you cut yourself, so we need to get platelets over to that location. Hey, you just had a nice meal, so now we need to get enzymes to be released in your digestive system. Hey, you're burning more calories because you're exercising, so now we need more ATP. So the brain figures out how to respond to the sensory division, and once it knows the task that needs to be done, it's going to send it over to the motor division of the peripheral nervous system to get that task completed. And the motor division, as you can see, it's going to carry out the signals from the central nervous system. It's going to be responsible for producing the response. It's going to be responsible for producing the response. It's going to produce the response that you require. And once again, we're going to have two divisions. We're going to have the somatic motor division, which will once again concentrate on getting things like contractions in your skeletal muscle and focusing on reflexes, okay? So if you see that you're swinging your arms, your legs are moving faster, you're doing the reflex of, hey, you hit your hand or you touch something hot, you're pulling it away, that is all a response. That's part of the somatic motor division. If there is a response that's internal, like maybe we are secreting enzymes, maybe we are producing different hormones, maybe we are moving around our cardiac and smooth muscle, well, that is going to be a visceral response, because once again, visceral means internal. So your visceral motor division will take a look at all the response, all the effects that are required for your internal glands, your internal cardiac and smooth muscle, basically everything that falls under the non-voluntary response. Now, one quick thing. In internal responses, so the visceral motor division, it turns out that those responses can either be something that is arousing, so you make it beat faster, you make it respond faster, you increase the secretion, or you slow it down. The heart starts beating slower, your breathing rate starts going slower, you stop the secretion of your enzymes. So everything's halted and kind of calming down. So what we see happening is when you have an arousing effect, we call that the sympathetic division. And when it's calming, that's the parasympathetic division. So for instance, whenever you feel like the fight or flight, so maybe someone made you angry, someone stole your parking spot, and you just feel yourself getting angry, that's a fight or flight response. And you feel your heart beating faster and your breathing starts to increase, that's an example of a sympathetic response because everything's increasing. And it's a visceral response because your lungs and your heart are all located internally. Now that's why they tell you when you get upset, just stop, take a few deep breaths, and with that you feel yourself calming down, you feel your heart rate slowing down, you feel your breathing going back to normal. That's the parasympathetic division kicking in and now just reverting you back to your normal, relaxed, homeostatic state. So they will work together. If the numbers go up, sympathetic. If the numbers go down, nice and calming, parasympathetic. As you can imagine, you will also have neurons that will specifically be dedicated to the different systems, right? So here we have an illustration when we take a look at our neurons, so the cells that we find within the nervous system. And what we're going to see is we're going to have three different types of neurons. You have the neurons that will focus on the sensory system. So they'll be the ones that will monitoring any type of external features within the body as well as internally, right? They're able to detect any changes. Are you, summer where it's cold? Is it raining outside? Did you cut yourself? Are you hungry? Did you have lactic acid because you're using your muscles more than normally? This is all being detected by sensory neurons, which we also call afferent neurons. Those neurons are part of the peripheral nervous system because remember, that's where your sensory part comes in, right? In the peripheral nervous system. Now look what's going to happen. The sensory system, this is me looking at my drawing, is going to detect any type of stimulus and it's going to go ahead and communicate that with the central nervous system. And in the central nervous system, we come across our second neuron. This is called an interneuron, as in it sits in between. The interneuron sits in between the sensory and the motor response. It is the only neuron that's located in the central nervous system, so it's part of the brain and the spinal cord, and this is the most common neuron that we have. Look at that, 90% of our neurons are interneurons. So they're able to receive the sensory information, they're able to figure out what needs to get done, and once they know what the effect needs to be, they will communicate it back to the peripheral nervous system, and they will communicate that with the third neuron, which is going to be our motor neuron. And the motor neuron is the one that comes in direct contact with whatever muscle or gland needs to be stimulated. So remember your neuromuscular junction that we discussed in a previous chapter? This would be part of a motor neuron communicating with your skeletal muscle that it's time to contract that muscle. The motor neurons are also called efferent neurons with an E because they are effectors. So they cause an effect or a response. So these are the different types of neurons, sensory and motor, afferent and efferent, both in the peripheral nervous system. And then in the central nervous system, you have the most common neuron, which is your interneuron. Now here is a typical shape of a neuron. I know I've been doing my best drawing them. Obviously this is a much better illustration. And I just want to quickly remind you that in a typical neuron, you will have the cell body. That is where the nucleus is going to be housed. That's also what we call the soma. For those of us who have been studying up on our histology, you might have come across the term Nissl bodies. Nissl bodies are small little granules that stain within the soma. It's part of a staining process, and these are actually rough endoplasmic reticulum organelles. You can also see that you have a lot of different dendrites. So here are the dendrites. The dendrites are, of course, the ones that are able to receive signals and transmit it over to the soma. And you can have numerous of those. And then you'll have one and one only of the axon. Now, the axon is able to subdivide later down the path. You can see it subdividing here. But from the soma, there will only be one axon that will radiate outwards. And the axon is then the one that will go ahead and will be able to make synaptic connections, either a connection with another neuron or connection with effector or sensory cells, wherever we're looking for. So here, remember your little synaptic knob that we talked about, which was the end of the axon? That's the part that's going to be communicating with the next cell or the next muscle or organ, whatever you're working with. I also want to point out, and we'll talk more about this, is that notice how when you look at the axon, you have these little myelinated sheaths, these little sections that are wrapped up. This is called a myelin sheath. In a little bit, we're going to talk about exactly how it gets myelinated, and we're also going to talk about the fact that the axon likes to be myelinated because it actually speeds up their signal. So by wrapping up certain sections of the axon, it actually speeds up the signal that's being sent down the axon pathway. All right, now speaking of the axon, it turns out that we can have movement down the axon, which makes sense because we're basically communicating and making synaptic connection. When the movement is down the axon, so away from the soma, we call that anterograde movement. A for away, anterograde movement. And then we also see that there's movement that will go the opposite way. This is called retrograde. Retrograde means that you're moving up the axon, so you're going back towards the soma. This movement is usually only reserved for materials that have like vesicles, little containers that they would like to recycle, so they're bringing it back up. And there's also different pathogens, like for instance chickenpox, that will use this retrograde movement to kind of sneak up the axon and into the soma where the virus can kind of lay dormant. We have different types of movements, as you can see on the slide. It talks about fast and slow movement. I'm really more concerned by you knowing that when you see anterograde, that it means that we're moving down the axon, and when you see retrograde, it means we're moving back up. And also at the bottom, I did write down the two most common motor proteins that we see in this transport. For anterograde, that's the kinesin motor protein. So this is going to be the protein that literally makes like a little vesicle that can be used to carry the products down the axon. And then the protein that does the work if you're doing retrograde, that's the dynein motor protein. So once again, it's just providing the movement option or little vesicles to either move anterograde or retrograde. Anterograde is for the kinesin, retrograde is for the dynein. Here is another interesting fact about the neurons, or I should say the nervous system, right? So when we discussed this in the histology section, chapter 5, I said to you that when it comes to the nervous system, you basically have two cell types. You have your neurons, which we've been talking about a lot, dendrites, soma, axon, all that other good stuff. And then you also have your glial cells or your neuroglial cells. The neuroglial cells outnumber the neurons about 1,000 to 1. They are what we like to call the housekeeper cells of the nervous system. They are there to protect the neuron. They are there to do that little myelinated sheath that I was talking about. They are there to kind of make sure that there's no injury. They have many different functions. So what I want to do is I want to talk about the different types of glial cells that we come across in the nervous system. And of course, as we do different types of cells, we want to focus on what their ultimate function is. So let's go ahead and start off with the oligodendrocytes. The oligodendrocytes. They are going to be doing myelination, so they're going to form that little myelin sheath, that little wrapping on the axon, but they're only going to do it in the central nervous system. So the oligodendrocytes are an example of a glial or neuroglial cells that form myelinated sheaths, so wrapping around the axon in the central nervous system. It's important to point out central nervous system because in a little bit I'm going to show you the cell that does that work in the peripheral nervous system. So the oligodendrocytes are going to do the myelination in the central nervous system. And as you can see right here, it says it insulates it, so it speeds up the signal conduction. We're going to talk more about that in a second. The epidermal cells are the next glial cells. They are going to be the ones that you find within the cavities of the brain and the spinal cord. Their function is to produce the cerebral spinal fluid. So they will produce cerebral spinal fluid, CSF. That, of course, is the liquid that will bathe the brain and the spinal cord. Microglial cells are a form of white blood cells. They are modified macrophages, so white blood cells that can phagocytose, bulk eat any type of pathogen or foreign entity. They will be exclusively monitoring the brain tissue, and they will usually be, if they appear in high numbers, they're usually an indicator that there has been some type of trauma or damage or infection happening within the nervous system. Your astrocytes are the most abundant glial cells. They get their name because they kind of look like stars. The astrocytes will have many different functions. Their main function is to form the framework of the central nervous system. So they, in essence, monitor many different aspects of the central nervous system, including the blood-brain barrier. They have these little extensions called perivascular feet, which allows them to interact with the blood vessels in the brain, creating the barrier, which of course is essential because we want to make sure that there's no type of pathogens accessing the cranium cavity easily, because that of course could be detrimental to the nervous system. When the astrocytes are damaged, your brain has a really hard time clearing them out. So instead what we see is that they start to form this hardened mass called sclerosis. I bring this up because there's a lot of research being done now to see that individuals who have high amounts of sclerosis, so these damaged, hardened spots, they are usually more prone to develop diseases like dementia or Alzheimer's, just because it interrupts the synaptic connection between the healthy tissue and the brain. Schwann cells are next. Now, Schwann cells are going to do myelination, just like the oligodendrocytes, but they are going to do myelination in the peripheral nervous system. So the Schwann cells will do myelination in the peripheral nervous system, so anything outside of the brain and the spinal cord. And last but not least are our satellite cells. Satellite cells are going to be important because they are going to allow for electrical insulation, as well as regulate our chemical environment. And of course, this is important because our neurons are able to be electrically stimulated. So when they send off that nerve signal, we want to make sure that it travels properly, and the satellite cells will partially help with that as well. On the illustration that's inserted towards the bottom, you can go ahead and you can see we have our little neurons right here. Now notice that you have your oligodendrocytes. They are myelinating parts of the axon. So that means that this picture is part of the central nervous system, right? Because the Schwann cells do the same job, but they do it in the peripheral nervous system. We also see our little astrocyte right here, looking like a little star. And notice those little perivascular feet, how it's interacting with the blood barrier. Here are your epidermal cells. And then, of course, we have our microglial cells right over here. There you go. Microglial cells are, remember, an extension or an example of a white blood cell. It's a modified macrophage. Please take a few minutes to kind of review the different glial cells, be able to explain the function of all 6 that have been mentioned. Now, let's talk a little bit more about myelination. So myelination, or forming a myelin sheath, is basically when we wrap an insulating layer around the axon, also known as the nerve fiber. And as I pointed out to you in our previous slide, it's going to be the task of 2 different glial cells. The oligodendrocytes are going to do it in the central nervous system, and then your Schwann cells are going to do it in the peripheral nervous system. Both of them will provide the wrapping. They'll just do it at 2 separate locations, and this wrapping is mostly made out of fat. It's 80% lipids, 20% protein. So under the microscope, the myelination is going to appear white. And that is why for some of us who've been kind of studying histology a little bit more, or those of us who are more familiar with the nervous system, we know that there's regions that we call white matter versus gray matter. While the white matter is the section that's myelinated, the gray matter is unmyelinated. Not all your axons are myelinated because that would take up too much space within the cranium and the body, so only selective axons will get the myelinated treatment. But keep in mind that this myelination is done by the oligodendrocytes in the central nervous system and the schwann cells in the peripheral nervous system. And the point of myelination is to speed up the signal that travels down the axon so it can get to the destination faster. Now, there are some differences as to how the cells go about myelinating. So here on your PowerPoint, it says that in the peripheral nervous system, when we're taking a look at our schwann cell, we see that the schwann cell itself will wrap a section of the axon, and then the cell will sort of become trapped in the outer layer, which we're going to call the neurolema. So take a look at the picture that's inserted, and you can see the little axon right here, and then the wrapping that's around it, that's the myelination. So that's mostly lipids, a little bit of protein, and the outer layer of the myelin sheath, that's called the neurolema. That's where my schwann cell is going to be stuck because it's going to kind of get trapped in its own myelination. In the central nervous system, we don't see that, and that's because the oligodendrocytes, they are able to basically myelinate several fibers at the same time. So they will almost have like an extension where they're able to connect with different axons at the same time so that the cell doesn't get stuck in any type of outer layer. Keep in mind that the myelination occurs only in certain sections of the axon. So here's my little axon. So we'll myelinate a piece, and then we'll leave a piece blank. We'll myelinate another piece. And the blank pieces, the little gaps, those are going to be called the nodes of Ranvier. So those are like the empty spots where the axon is bare. I have a better picture coming up, I promise you. So here, take a look at this picture right here. So we have our myelination in our peripheral nervous system. Here's my little schwann cell. And notice how the schwann cell will find its axon, and it will start wrapping it around, follow the red arrow. And when it gets to its outer layer, that's when it forms the neurolema, because the schwann cell is right over here. It's stuck. On the other hand, if we take a look at our oligodendrocytes in the central nervous system, notice how you have the nucleus of the cell, and then notice how the cytoplasmic content is split, and it's making connections with 1, 2, 3 different axons at the same time, and it's making the myelination, but it's not getting stuck in any of it. So there's no neurolema occurring in the central nervous system. This myelination process will occur during fetal development, but it will really ramp up during the first months and few years of development. And that's one of the main reasons why infants and babies, if they need to make sure that they're getting proper amounts of lipids, fats in their diet, because that fats are needed for myelination. So the joke is always no skim milk, right? Give the babies whole milk when they're transitioning, because you want to have a high fat content that can be used for myelination. Here are just some illustrations that I found from Google Images. Obviously, I know a lot of you are very visually inclined, so I figured you might enjoy them. Here on the left-hand side, you see an oligodendrocyte connecting with an axon, and you can see it myelinating. And notice how it has several extensions coming off its cell body. And then on the right-hand side, we have two different axons, well, two different neurons, I should say. And the little blue spark is meant to stimulate the electrical signal. So that's the neurotransmitter coming down the axon. And it's just highlighting the fact that if the axon is myelinated, so it has the wrappings around it, that signal can proceed at a much faster rate. Look at them go. And then here is a better picture of the nodes of Ranvier, as I was mentioning to you before. So those are the little gaps that sit between the myelination. And then I do want to kind of point out that on this picture right here, it's showing you your little schwann cell, and you could see your axon, and you could see the myelination happening. And I do want to point out that I said to you that the myelination is really important because it's obviously going to increase the speed of the nerve signal. So the presence of the myelin will definitely contribute to that. And another item that will regulate how fast the signal can pass down the axon or the nerve is going to be the diameter of the fiber. So how thick the actual nerve is. A small nerve will be easily to stimulate, which means it will travel faster, whereas a larger nerve will take a little bit more stimulation, so it will slow down the signal. So the two factors that will determine the speed of the nerve signal at the end of the day will be the diameter of the fiber and the presence of the myelin. Alrighty, this chapter is relatively straightforward, and it's actually one of the shorter chapters, so I think we're going to call it quits on part 1 right over here, and then we'll pick up for part 2 in a little bit, and we'll talk about things like action potential and exactly what happens when a neuron gets excited and how it sends its electrical signal down the axon. Alright, as always, questions, comments, concerns, feel free to send me that email, and we shall chit-chat soon again.